citrate synthase. Acetyl-CoA oxaloacetate citrate is catalyzed by CoA. Acetyl-CoA oxaloacetate citrate produces citrate. Acetyl-CoA oxaloacetate are condensed to produce cis-Aconitase. Citrate cis-Aconitate is catalyzed by cis-Aconitate. Citrate is dehydrated to produce the intermediate compound. Removed. Is-HO added or removed in the citrate cis-Aconitate reaction. Aconitase. Cis-Aconitate isocitrate is catalyzed by Isocitrate. Cis-Aconitate is hydrated to produce added. Is-HO added or removed in the cis-Aconitate isocitrate reaction. Isocitrate dehydrogenase. Isocitrate alpha-Ketoglutarate is catalyzed by alpha-Ketoglutarate. Isocitrate in NAD NADH leads to NAD NADH. Isocitrate alpha-Ketoglutarate half reaction. NADH and CO. What is produced in the isocitrate alpha-Ketoglutarate reaction? Alpha-Ketoglutarate dehydrogenase complex. Alpha-Ketoglutarate succinyl-CoA is catalyzed by succinyl-CoA. Alpha-Ketoglutarate and coenzymes leads to the energy-rich oxidative decarboxylation leads to the energy-rich NAD and CoA. What are added into the alpha-Ketoglutarate succinyl-CoA reaction? NADH and CO. What are removed from the alpha-Ketoglutarate succinyl-CoA reaction? Succinyl-CoA synthase. Succinyl-CoA succinate is catalyzed by CoA and GTP. Succinyl-CoA succinate produces what two things? Succinate. Succinyl-CoA undergoes substrate-level phosphorylation to produce succinate dehydrogenase. Succinate fumarate is catalyzed by fumarate. Succinate and FADH2 FADH produces FAD-FADH. Succinate fumarate half reaction. Fumarate hydratase. Fumarate malate is catalyzed by malate. Fumarate is hydrated to produce malate dehydrogenase. Malate oxaloacetate is catalyzed by oxaloacetate. Malate and NAD NADH produces NAD NADH. Malate oxaloacetate half reaction. Which step does CoA get removed from? Acetyl-CoA oxaloacetate citrate. Which step does HO get removed from? Citrate cis-Aconitate. Which step does HO get added into? Cis-Aconitate isocitrate. Which step does NAD get added into, and CO and NADH get removed from? Isocitrate alpha-Ketoglutarate. Which step does CoA and NAD get added into, and CO and NADH get removed from? Alpha-Ketoglutarate acetyl-CoA. Which step does CoA get removed from? Succinyl-CoA succinate. Which step produces GTP? Succinyl-CoA succinate. Which step produces FADH from FAD? Succinate Fumarate. Which step does HO get added into? Fumarate Malate. Which step does NAD NADH occur? Malate Oxaloacetate. Purpose of Krebs cycle. Provide reducing power. Overall function of Krebs cycle. Oxidize 2 carbon atoms of acetyl-CoA to CO. Produces how many CO? 2. Produces how many NADH? 3. Produces how many FADH? 1. Produces how many ATP, GTP? 1. Name a silly mnemonic to help you remember the Krebs cycle. Can I keep selling sex for money, officer? Irreversible steps of Krebs cycle. 1, 3 and 4. Only step with ACC bond formation. Step 1. In what conformation does citrate synthase have a binding site for acetyl-CoA, and how is this accomplished? Closed conformation, triggered by binding oxaloacetate. Step that involves a double bond. Step 2a. What part of aconitase catalyzes the removal of water from citrate and the addition to cis-Aconitate? Iron-sulfur center. What facilitates the electron removal of the isocitrate dehydrogenase reaction? Mn2. What enzyme has the same coenzymes and mechanisms as PDH complex? a-Ketoglutarate dehydrogenase complex. In the hydration reaction, blank is added before blank. OH. Oxidation of malate oxaloacetate is thermodynamically unfavorable, so why does it occur? Because oxaloacetate is so low, having been constantly used to make citrate. What happens to the carbons of the acetyl group from acetyl-CoA? Oxidized to CO2. How many ATP are ultimately produced from a molecule of NADH and FADH2? NADH equals 2.5 FADH. The two NADH from two turns of this reaction may end up in 3,5 ATP depending on shuttling mechanism. One, 3-bisphosphoglycerate, 3-phosphoglycerate, 3 points at which CO2 is given up. Pyruvate to acetyl-CoA, isocitrate to a-Ketoglutarate to succinate-CoA. Ultimate net ATP formed from metabolism of one glucose molecule. 30 to 32. 3 differences in linear citric acid cycle of anaerobic bacteria. 1. Second entry point. 2. No a-Ketoglutarate dehydrogenase. 3. 2 exit points. What are the 2 exit points of anaerobic bacterial citric acid cycle and what do they lead to? A-Ketoglutarate and succinate-CoA lead to biosynthetic products. What is the second entry point of anaerobic bacterial linear citric acid cycle? Pepper pyruvate is carboxylated to oxaloacetate. Energetic gain from linear citric acid cycle. NADH. Explain the amphitabolic pathway of the citric acid cycle. Contains anabolic and catabolic processes. What are anaplerotic reactions of citric acid cycle? 1. Reactions that replenish intermediates. 2. Use CO2 to turn 3C intermediates to 4C intermediates. 3. Reactions using enzymes. 3 ways citric acid cycle is inhibited. 1. Regulation of pH complex. 2. Substrate availability. 3. Competitive feedback inhibition among intermediate. 4 things that inhibit pH complex and 4 things that stimulated pH complex. Inhibit ATP, acetyl-CoA, fatty acids, NADH, stimulate AMP-CoA, NAD, circuit 2. 4 things that inhibit citrate synthase and 1 thing that stimulates it. Inhibit NADH, succinate-CoA, citrate, ATP, stimulate ADP. 2 thing that inhibit isocitrate dehydrogenase and 2 thing that stimulate it. Inhibit ATP, NADH, stimulate circuit 2, ADP. 2 things that inhibit and 1 thing that stimulates the a-Ketoglutarate dehydrogenase complex. Inhibit succinate-CoA, NADH, stimulate circuit 2. How do animals gain energy from fat? Degrade lipids by B-oxidation to acetate which enters citric acid cycle. How do plants and other microorganisms gain energy from fat? Glycoxylate cycle. Where does glycoxylate cycle occur? Glycoxysome. Why doesn't the glycoxylate cycle contain dehydrogenases? Cannot reoxidize NADH. In glycoxylate cycle, how is malate converted to oxaloacetate? Transported into cytosol. Two enzymes of glycoxylate cycle not found in TCA. Isocitrate lyase malate synthase. Is the phosphorylated or dephosphorylated form of isocitrate dehydrogenase active? Dephosphorylated.